Hello, everyone. I'm S.Hoban. I'm at the Morton Arboretum, which is a botanic garden in Chicago. And yes, really happy to be here to speak to everyone today. Should I share the screen and start the presentation?
Yes, please go ahead.
Okay. Okay, can everyone see the first slide?
Yes, we can.
Okay, good. And it's just that, yes? Okay, I will begin. Thank you again. So I'll talk to everyone, to you all today about conserving genetic diversity ex situ in botanic gardens and seed banks, both how much to safeguard and how to do it. There will be kind of 4 segments to the talk, short background, and then I'll talk about some studies that I have done over the past 5 years on both rare and common species, starting with rare species, but then also talking about common species at different spatial scales. How do we conserve their genetic diversity ex situ? And then I'll close with a few thoughts about the next series of projects that need to be done to build on this work. And if people have questions throughout the talk, you can of course unmute your microphone and ask, or you can use the chat box, or we can just save those for discussion at the end. But please feel free to interrupt if you like. We're all aware of the challenge of conserving biodiversity to keep all of the pieces of the ecosystem and the species and the genetics, even if we don't know precisely how useful each of those pieces will be in the future. So they might be used for breeding, for reintroduction, for scientific study, etc. So the best we can do is usually to gather a representative sample of the genetic trait and ecological diversity across the species range, and manage that for the long term. So that means potentially for multiple generations, just like you would do in a zoo, potentially multiple generations in a botanic garden, and for a seed bank, also potentially planning how often you have to go back to sample more seed. So I've been focused, and what I'll talk about today is primarily the problem of how much to collect and how to do it mostly spatially. The balance, of course, is between efficiency. We have limited resources, time, space, personnel, especially in botanic gardens. But we want to be as effective as possible to preserve all of the genetic variation. People started working on this topic at least as early as the 1970s. This was a time of agricultural homogenization and industrialization, when agriculture was losing the traditional varieties that people have developed and maintained for thousands of years. So our rich biodiversity heritage, and so scientists wanted to conserve these in seed banks. And they recognize that there's, of course, limited size to seed banks. And so the first scientists that I'm aware of who worked on this, they took this as a simple probability problem. They said there are genes, genetic variants at a given frequency, alleles at a given frequency, and we're basically trying to capture the rarer of those alleles. So they used equations like this one to come up with simple recommendations that should apply to most species if the species fit kind of the classic genetic model of a population and have normal allele frequencies. And so this should capture all but the most rare alleles, this recommendation of 50 samples per population. And for common species, they recommended visiting about 50 populations throughout the range. This recommendation has been used by seed banks and botanic gardens for really ever since, for decades. But what this simple recommendation neglects is that species differ in their biological traits, and especially traits which are known to affect genetic diversity distribution on the landscape and across the range. So this is seed dispersal, pollination mechanism, size, longevity or lifespan, and range size and geographic fragmentation of the range. All of these traits will determine how much genetic diversity is within and among populations, and therefore how well a given sampling strategy, which might be number of populations, number of trees, and number of seeds per tree. And so my first work on this topic was when I was a postdoc, and I found that because of these traits, theoretically, we should tailor our sampling strategy to the target species. That some species, especially those that are selfing, have low seed dispersal, will need up to 5 times as large of a seed bank. The main message was that 50 samples is a good guideline, but it can be tailored to particular species. So now I'm going to talk about what I've done since then, especially with some empirical work. How much have we conserved in botanic gardens? Can we put a number on this? How much should we be conserving in botanic gardens? And then I'll talk about sort of my interest in developing practical rules or recommendations for when we don't have genetic data. And then I'll talk about what are we trying to conserve anyways, because actually the philosophy of conservation is very important. The first set of results regards 11 rare species across 5 genera. This was a project led by the Montgomery Botanical Center in Florida, and these are some of the participants. How do we determine how much we have conserved? It's a pretty basic process. We go out into the wild, that's the picture in the upper left, and we get tissue from all, these are rare species, so we can try to collect all or most of the living individuals and at least to visit all of the known populations. We then go to botanic gardens. We use BGCI plant search and other contact lists to find all the botanic gardens that have these target species. And they send us leaf samples. Then we get DNA out of the wild samples and the garden samples. And we simply compare the genetic diversity in the wild to what is conserved ex situ. And this takes a fair amount of work in terms of finding all the populations, visiting them, and coordinating among, usually these samples are spread among several dozen botanic gardens. These are the 11 species, and how many samples, so how many trees, how many living individuals are in botanic gardens today. So some rare species have very few individuals, and some rare species have hundreds of individuals in botanic gardens. So this is important even just to consider the range of population sizes in botanic gardens. But this is how much genetic diversity we found in those samples. The green arrows show those species that have about 95% of their genetic diversity conserved, and the red arrows show species that have 70% or less of their genetic diversity conserved. So some species, the big message is some species are conserved very well, but probably more species are not meeting what we would hope to have as our conservation goals. There is a pretty clear relationship between the number of plants in the botanic gardens, that's the x-axis, and the amount of genetic diversity conserved. So each of those points is one of our species in the study. As you get more individuals, you get more genetic diversity. So that's a big recommendation, and it's something we would have known a priori. But I point out a couple examples here where we have 2 species. One species has actually been conserved much larger numbers in botanic gardens, but has lower genetic diversity conserved. Here also, one species has twice as many samples, but less genetic diversity. And here also, 2 species with basically the same number of individuals in botanic gardens, but one of them has twice the amount of genetic diversity as others. So what does this tell us? It tells us that it's not just about the number of individuals you keep in the garden. Of course, more is better, but it's also about how you collect. So that leads me to my next question. Okay, so as a seed collector myself, I know that seed collectors often go to one or a few accessible populations, and they don't sample throughout the population. They get accessible individuals, those that are producing a lot of seed. It's the reality of sampling. But if we could go to a population and sample randomly throughout the species range, that would be optimal. So this next little bit is about, if we could go out randomly, how much should we be sampling at an absolute minimum? So we take our wild genetic data set, these trees shown here, and we resample it for different numbers of trees all the way up to the total number of trees. Every time we do that, we calculate how much genetic diversity is conserved in that sample. So this is the x-axis, is the number of individuals we resample from our wild genetic data set, assuming perfect random sampling. And the y-axis is how much genetic diversity in each of those resamples. Then we can take a threshold, say our goal is 95% of the genetic diversity. We intersect with this line here, and we drop down this vertical line, and that tells us how many individuals are needed to get 95% of the genetic diversity. So I did that for all the species in our study, these 11 species, and we see that from the species that requires the least is only 24 samples. So that's lower than the 50 samples recommended by Marshall and Brown, but some of the species require 70 or 80 or more samples, which is of course higher than 50 samples. So there is a range among species, probably based on their biogeographic history, their recent bottlenecks, the amount of genetic diversity and population fragmentation. All of that will contribute to how many samples are needed. The main point is that this complements my previous predictions that yes, if possible, we can optimize our sampling to the target species. So yes, we should sample differently for different species if we know about their genetic diversity. And unfortunately, it doesn't really correspond to the genus. We were hypothesizing that species within a genus which share some traits should be sampled similarly, and they don't really cluster together in this list, except for Quercus. Of course, this work should be expanded to many more species to verify that. So if we talk, so far we've talked about how much genetic diversity is conserved today, and how much we should be keeping. If we put those 2 points together, we can then ask the question, how much better could we do if we could sample better in the wild? So this is Quercus boyntonii. It's a endemic species to really just a few dozen kilometers area in Alabama, maybe 50 kilometers. Current collections around the world at maybe 10 botanic gardens, 77 individuals, capture about 70% of the wild genetic diversity. But if we could go back into the wild populations and make a new collection, sort of design it optimally, those 77 individuals could get 94% of the genetic diversity. The main point here is that we can, with the same number of individuals in our seed banks or botanic gardens, get more genetic diversity than we currently have, just by improving sort of our sampling among populations. So we call this the genetic conservation gap. In this species, it's about 1.35 times as much benefit for the effort that we spend. And that's really a lot. If we look at across all the species, it's about 1.4 times as much genetic diversity, and as much as twice for some species, without increasing the size of the collection. And this is really, I mean, in conservation, that's a really big improvement. 30 to 40 to 50, even to 100% improvement. Often in conservation, we're talking about improvements of a few percent. So this is really a large increase we could make. Of course, it's hard to collect optimally, and we can revisit that in the discussion. Are there any questions at this point? I've sort of wrapped up my section on the rare species, and we'll then move to common species, but are there questions so far?
Perhaps I could be the bold one and ask a question.
Nigel, thank you. Please introduce yourself.
Oh, so my name is Nigel Maxted. I'm Professor of Conservation Genetics at the University of Birmingham. So my question is really, you're talking about the ideal number to capture genetic diversity, but there is a difference between this ideal number and the practical number that you might want to have if you were running a gene bank. And so the difference, in my view, is that obviously you want the material to go out. So you must have enough collections to be able to supply demand as well as conserve overall genetic diversity. So that's the first question. And then the second one is the assumption is in this so far that you're just using ex situ conservation. But if you had a combination of both ex situ and in situ conservation, how would that impact on your collecting strategy?
That's really good. The first question I will get to towards the end of the talk. I have a small project on how much we increase the size of the seed bank if we are either using it or if there's some threat of loss, so how much duplication we need. So we will get to that. But the second question is something I have not worked on and I don't have any ideas about other than to say, perhaps, yes, if you have some in situ populations that are very secure, you know they're stable and protected, maybe you don't need to sample them, and you only want to sample those that are really highly threatened or likely to be extirpated. Yeah, you could just focus on that as your subset. I think you could use these same methods and just focus on those more threatened populations as your target. But I haven't worked on that yet.
Thank you very much. Are there any other questions at this point? Then please, you can continue.
Okay, thank you. Okay, so this was a series of projects I just finished talking about over the past few years on these rare species. All of those were rare species with maybe 2 to 10 populations, all of them with less than 1,000 individuals living in existence for the whole species. I also worked on some common species, in particular this one with Claire Trivedi at the UK National Seed Tree Project. Fraxinus excelsior. We focused just on the populations in the UK, which is already a lot. It's I don't have the statistic here. It's in the millions of trees. It's their second most common broadleaf tree in the UK, and we're talking, I mean, thousands of individual populations scattered throughout the UK. They've been collecting seed for the past number of years, and now have over 1 million seed. They have conserved over, in our estimate, which is based on some genetic data, but also models, because we can't get genetic data on millions of wild trees. So we had to do some extrapolation and estimates. But we estimate that they've conserved over 90% of overall diversity. And we also focused on what we call locally common alleles. These are alleles only present in one or a small number of populations, but not many populations. And they've actually done really well at those locally common alleles because they have a widely distributed, really nice sampling strategy. I should have put a picture, but it's quite well distributed spatially. So we estimate that was our first question, was how much is in the seed bank today? Can we estimate that? And then I started to think they do have over 1 million seed. Of course, like Nigel just mentioned, much of that seed needs to be used for experiments, restoration, etc. But I thought about, what about when should a seed sampler stop if they have very limited resources? You look at these kinds of curves, which show the accumulation of genetic diversity on the y-axis as you add more seeds or as you sample from more maternal trees. And they flatten out, all of them flatten out at some point. So I used a method to look at when this curve gets pretty close to flat, and we can define what pretty close to flat means. A tangent to that line, some increase in genetic diversity for adding more effort. And for this species, I estimated that flat point is about 30 populations, about 30 trees or a little less, and about 15 seeds per tree, which is very different than they are sampling. They have actually more populations, but fewer trees per population because it's hard to find the trees, and many, many, many more seeds per tree because, of course, it's very easy to get Fraxinus seed. You're talking many thousands of seed per tree. So their actual sampling does differ from, the optimal or most efficient sampling. Not saying that this is the best sampling, but it's interesting to compare that to what's actually being done. And then I looked zooming out spatially how to sample. You can imagine an organization, a seed collecting organization, which has either more space in their seed bank or is more limited by their funds for travel. Maybe they're limited by some political borders and they can't visit all the populations. There are definite limits on both space and travel. So these are two options, A and B. Shows option 1, you visit fewer populations and get more trees per population. Option B, you visit more populations, but very few trees per population. You take the same number of seeds per tree. One of these, of course, involves much less travel, and that's the first. But it takes up more space. So it takes 250 trees that you're sampling, so a bigger size of your seed bank. This one's only 90, so less than half, almost a third as small. Both of these get the same amount of genetic diversity. So the main point here is you can get the same amount of genetic diversity with very different sampling strategies, whether you want to commit more resources to travel or to space in the seed bank. So I think that's useful practical knowledge for the seed collector. Also, in terms of spatial sampling, this is within populations. For those of you who've done seed collecting, you're probably very familiar with when you encounter a population, you're time limited. You maybe sample the 21st trees that you can find, or maybe you take a transect through the population. So I compared these strategies within populations. The same concept applies to among populations across the species range. This point sampling is this pink one here, and it's not quite half as much as the sort of grid or random sampling, but you can see it's significantly a lot less genetic diversity. Again, this is genetic diversity on the y-axis. Much less genetic diversity than a more grid or random sampling. So it's important that not only can we say, yes, it's important to randomly sample, but this is how much important. We can quantify that. So these are a few things especially about spatial sampling. I'll talk next about sort of the philosophy of conservation, and I'll revisit Nigel's question about duplication in the seed bank. But any questions on these couple of results about mostly about Fraxinus?
Apparently not, but I found it really interesting. I think I would like to come back because it could be relevant also in defining our in situ conservation strategy. But and I know that it's just a mathematical computation behind, but I think, in designing and our minimum number of population conserve in situ, then we often struggle with this number, especially because we have many populations that have very, very few trees. So for us, it was a big challenge defining when a population was worth to be conserved, even if it was having very, very few trees. But please go ahead.
Okay, thank you. So then now I'm really starting to think about basically what Nigel had said. How are we using these seed banks? What's the goal of the seed bank? How is it going to be used? Or botanic garden? So the first question is, what kind of genetic diversity do we need? Some alleles are very rare and hard to capture. Do we need to protect them all? The second question it relates to Nigel's question. How much duplication do we need? I've been thinking more in terms of protecting against disaster. Botanic gardens and seed banks are subject to fires, hurricanes, pests, and disease for botanic gardens. And to protect against these, we have to increase the size of our seed bank. Basically, we want to capture more than one copy of every allele. Everything I've told you so far assumes our goal is to capture one copy of every allele. Maybe we increase that to 5, 5 copies, we have to increase the size of the seed bank by almost a factor of 5, not quite, but close to it. If we want 25 copies, we have to increase the size of the seed bank by about 20 times, which is a lot. And the same goes if we're using those up for restoration or scientific study. So this chart will summarize those two things. Towards the right are the rarer alleles, and towards the bottom are more duplication, more copies of each allele. These are the kind of spread of recommendations within each of those categories. This is due to the traits. So this is the first part of the talk. I talked about species need to be sampled differently because of their traits. 2 to 4 to 5 times difference among species. But this is due to the philosophy of conservation, the type of genetic diversity, and how many copies we need. So that's a lot more. That's a factor of 64 between that lowest amount and the highest amount, what we're doing with the seed. That's what I want to work on next, is the likelihood of loss and the uses of the collection, and also the importance of rare variants. This is harder. I don't have a good answer on that, but I think it should be very easy to calculate the likelihood of loss based on past disasters and the uses of the collection. So I'm going to just talk about a few next steps and then finish. This is the first next step, is the philosophy. The second next step is about adaptation. Possibly with U4Gen or other projects, you've looked at maps like this, where you place your populations, these are circles, on a map of geographic, ecoregions or other geographic variation. The white circles here, this is a Quercus georgiana in the southeastern US. The white circles are where it exists. The black circles are where we actually have seeds sampled from in our seed banks. So there are large parts of the range and distinct geographic regions that are not sampled for our seed banks. So the first easy recommendation is to get more adaptive diversity, more ecological diversity. We have to visit these unsampled ecological regions, which might not show up as neutrally genetic different, but probably have adaptive differences. The next steps beyond that, I'm thinking about like how useful are the collections in our seed banks and botanic gardens, how well are gardens coordinating with each other, but also if we have one tree in a botanic garden and it has no trees to exchange pollen with, maybe it can't produce seeds. Maybe there's 2 trees, but they are siblings, so anything from them would be inbred. Some trees are too young or maybe too old to produce much seed. And some trees might be hybridizing with other trees, and we maybe not want that seed for conservation or restoration. So that's another thing I want to think about and work on, is how useful are the trees and seeds in ex situ collections. And then even beyond genetics, as a botanic garden, we are trying to get not just genetic diversity, but also phylogenetic diversity, horticultural value, all the rare species, ecological diversity. We have 4 to 6 goals or dimensions we want to achieve as a garden, and I'm thinking about how to balance those different goals. So I want to thank the many people who I've worked on these kinds of projects over the years, and the people who funded them. And to summarize, I told you that we have, for rare species that we looked at, 40 to 95% of genetic diversity conserved. These species have had a lot of work done on them, so these are probably actually optimistic numbers. Most rare species are probably lower than this. We can optimize our sampling to the species based on its traits. We can increase the amount of genetic diversity without actually putting more trees into our collection if we do more optimal sampling. We can look at these kind of general rules about stopping points and spatially equivalent strategies to help the seed collector. And I think it's really important to think about what we're using our seed for in the future and changing our original seed collection to match that eventual goal. So happy to, if there's any more questions or discussion.
Thank you very much, Sean. Thank you for this very comprehensive presentation. I would like now to open the floor to all the people attending online. If there are any questions, you can easily raise your hands electronically or just switch on the microphone and you can start speaking. Nigel, I cannot believe you can be silent for so long.
But I don't want to embarrass myself by asking lots of questions. I need to get other people a chance. But perhaps I could start. I mean, you talked about this conservation strategy, having a conservation strategy. I think you weren't explicit about what that meant, but I presume what you mean is you have a very good picture of the spread of genetic diversity in a species, and then you deliberately partition it so that you know that, say, if you can partition genetic diversity into 10 clusters, you sample from all clusters. Is that what you meant? Or did you have another meaning?
Essentially, yes. That would be the optimal strategy, assuming you had some genetic data. So we would only be able to do this for a relatively small number of species for which we have that kind of genetic data. But yes, we would identify genetic clusters or populations and allocate sampling among them. If we didn't have that, then we could use these kinds of maps and assume that geographic or even spatial fragmentation, just make an assumption that corresponds to genetic differences.
So that would be your proxy. Your proxy would be eco-geographic diversity.
Yep, and what I want to do later this year is actually work on how good of a proxy that is. We know, of course, geographic differences, often cause genetic differences, but does that correlate to, if we use sampling designed on each of those two data sets or information sources, how much do each of those result in a seed bank with genetic diversity?
I'll let someone else have a go.
Thank you very much. Thank you, Nigel. Thank you, Sean.
Hello?
Hello, yes, Andrew.
It's Andrew Smith at Morton Arboretum. Yeah, Sean, very interesting. I wasn't clear from your maps whether the points you were showing were, they were maps of populations and the points were trees, or whether the points were populations. And in terms of collecting samples, if those points are trees, how important is it then potentially to record and isolate those seed collections by tree as opposed to population? Because I think common practice is you go to a site and all the seeds from various trees are lumped into one collection.
Yeah.
And depending on the randomness of germination and viability, you could end up with all the horizon plants potentially being from just one tree, even though the samples have been collected from, say, 9 trees. That must be a statistical problem here?
That is a really good problem. It's something I have not worked as much on the importance of segregating by maternal lines. But to answer your first question, yes, these are trees. These are trees within a population. The bigger circles are adults and the small circles are actually seedlings. I did simulated, but also real genetic data. So this is choosing trees within a population. But we did the same thing with Fraxinus excelsior, a very similar type of map, but at the population level. So these, you can picture it really either way. And the results were very similar for when we looked at Fraxinus excelsior across the UK. It was just as important to spread your populations out as it is to spread your trees out. I think, and maybe I'm open to other people's discussion, but I think whether to segregate by maternal line depends on your goal. For trees that are threatened by pests and disease, I would say yes, especially if you're using those trees for experimentation. It would be really important to know which trees might have genetic resistance in them, in that family. And it's also super important for the hyper rare species. So all of our botanic garden best practice for rare species says to segregate by maternal collection. And you have to keep track of that in your seed bank. So keeping them in a separate bag or a separate vial, because to prevent inbreeding later on, that's the main, I think the main goal of segregating by maternal lines is if you want to learn about the inheritance of the trait or go back to a given tree in the future if you identify a useful trait, or if you're trying to avoid inbreeding. I was just at a botanic garden conference just a few days ago, and they told me it's 4 to 10 times as hard, takes 4 to 10 times as much work in the field and in their record keeping to keep the maternal line separate. So it's a big challenge. But for those particular cases, I think it's very important. But I'd love to hear what you think also.
Well, I don't have a firm view other than, well, assuming ideally you would separate by maternal line, but I wouldn't have been surprised to hear that it was potentially 20 times more difficult to separate by maternal line.
Okay, thank you. Any other question?
Also okay for comments or suggestions of future work that needs to be done.
Yeah, Sean, have you written any of this up yet, or is this presentation the first sort of exposure to this thinking?
So most of this is published. I put some citations throughout, which you could go back on the video and look up. So most of this is published. This stuff about the future directions, this is published by my colleagues, Murphy Westwood and Emily Beckman. This is published. Yeah, most of this is published, except for the first things I talked about. So the rare species, it's in review right now, and should probably be published soon.
Okay, thank you very much. So if there are no other questions, then I will close this this web conference. Last chance to make questions. Then I would like to thank you very much, Sean, for your time, for giving this presentation. As mentioned, this presentation is going to be made available within the Generous Breed project, and then it will be broadcast during the workshop that we're organizing in a couple of weeks and in Paris. Thank you very much again. Thank you for your time. Thank you for everyone for attending.